In this video, we're going to be talking about glycogenolysis, which is the lysis, so the breaking of glycogen. And glycogen is a macromolecule, so a big-sized molecule made by many units of glucose. Even thousands of molecules of glucose bond with an alpha 1,4 type of bond. And glycogen also has branching points. It's got many of these points in which the bond is alpha 1,6. So the carbon 1 of a glucose molecule is bonded with the carbon 6 of another one. The fact that glycogen has a lot of branching points makes it so that the glycogen molecule ends up by having a lot of non-reducing terminals and just one reducing terminal, which usually bond to a protein which is used for the synthesis at first of glycogen from glucose. Now, as you can see in the picture over here, there's an alpha 1,6 bond, which is the origin of the branching points, and then all the other bonds are alpha 1,4, which the name has got to do with the conformation of the carbon and the oxidrylic group and the hydrogen bond to that specific carbon. Then the red molecule of glucose is a non-reducing terminal, which will then be attacked by enzymes in the real process of glycogenolysis. Now, just to give you an idea of how a glycogen molecule looks like, this is a picture I got from Wikipedia, in which you can see in the center there's a protein, which is glycogenin, which was used as primer for the formation of this macromolecule. And you can see all the branching points and the non-reducing extremities of the branches in this molecule. Now, glycogen is present mainly in the liver and in muscle cells. Nerve tissues has almost no glycogen. In the liver, it is about 8 to 10% of the weight of the liver, so that's about 100-120 grams of weight. But this happens only immediately after having eaten, so when the glucose levels in the blood are pretty high. After a period in which you don't eat, the glycogen lowers in quantity, and after one day of diet, it goes practically to zero. So in muscle cells and in the liver, what happens is that glycogen is broken down by glycogen phosphorylase, which is an enzyme which catalyzes a pretty amazing reaction. It breaks the sugar bond between the alpha 1,4 bond between the molecules of sugar, and what it does, it breaks it with an inorganic phosphate. So you end up by having glucose, just that it's glucose 1-phosphate. So it's already got a phosphate group, which will then be necessary for glycolysis and for the synthesis of new ATP. So it's actually saving energy by doing this. And then there's another enzyme which switches places to the phosphate group and moves it to the carbon 6. So as you can see in the picture over here, you're going to have glucose 6-phosphate, which can then enter the glycolytic process if it's in the muscles. If it's in the liver, the phosphate group gets removed and you have by another enzyme, and you have again glucose, which can go now in the bloodstream and reach other tissues which need glucose and which need energy in that moment, That's mainly brain tissue and muscular tissue. Now, this happens only when you need a lot of energy immediately and you don't have enough already in the bloodstream. So there are certain hormones which are going to catalyze the lysis of glycogen, and these are like glucagon, which is secreted by the alpha cells in the Langerhans islands in the pancreas, or also by adrenaline. It's going to catalyze the lysis of glycogen, and all other hormones which have hyperglycemic properties, which means they have the property to increase the level of sugars in your blood. And now the picture we're looking at is the picture of the molecule of glucose. You can see 6 atoms of carbon at every point of the molecule, and this molecule travels in the bloodstream and then is captured by the cells throughout specific transporters throughout their membrane, which are mainly they're called GLUT 1, 2, 3, and 4. And then there's also other types, and they have different properties according to the tissues in which they are found. And now we're going to see a molecule which is a protein. It's glycogenin, which, as I said before, is the primer for the synthesis of glycogen. Now, what a primer is, it's a molecule which helps the synthesis at first of the molecule, in this case of glycogen. And the reason for the existence of primers is that not all the enzymes can just work on a molecule out of nothing. They need a place to start on, and so that's the reason why primers exist. Now, the whole point of glycogenolysis is to increase the blood levels of sugar, to increase the amount of glucose in the bloodstream. So in this image you can see other ways to obtain the same result, and like all these ways work together to increase the level of glucose in the blood.